Хотела бы представиться вначале на русском. Мое имя Овчаренко Екатерина. Я студентка Сибирского государственного медицинского университета, 5-й курс, медико-биологический факультет. И что ж, на, собственно, данном заседании журнального клуба я представлю следующую тему. And if saying this all in English, I'm really happy to be here, to be present here and saying this all interesting things about innate immunity. So good day, everyone, and let's start with principles of innate immunity according to Jane Wayne's Immunobiology. So the first thing which is quite interesting in terms of innate immunity is another living beings, another organisms that can trigger our immune system's reactions. These living beings, these little creatures, can be described just simply as pathogens. They vary greatly in their size and the effects they cause in our bodies after they are after we meet them in our environment. So the littlest of them, the least of them, like the tiniest of them is, of course, viruses. There is lots of viruses that we can meet in our everyday life. Some of them are highly pathogenous and some of them are almost harmless to us. But nonetheless, viruses are here and they're pathogens, and they also can trigger our immune system's reaction, especially innate immunity reactions. Of course, the spectrum of pathogens also includes bacteria, which can be intracellular and extracellular. Also, there is archaeas and protozoas, and all of them as well interesting organisms as well. They are slightly bigger than viruses, and they also have plethora of effects on our bodies. Fungi and parasites are another groups of these pathogens that we can explore while talking about innate immune systems reaction. And it is also very important to notice that they are vary greatly in size, and because of that, they can include many different molecules and proteins in their structure that can cause different effects after meeting with our immune system cells and molecules. Of course, their size difference is not the only thing that it is important. Also, it is important to know which effect on our tissues, how they affect our tissues. Some of these organisms, some of these bacteria are quite commensal and they cause little host damage. And some of those pathogens, especially those which are present in this slide, they can cause quite a lot of harm. And of course, these relationships with human bodies are one of the topics that can be explored while talking about immune system reactions and innate immunity as well. Of course, while talking about pathogens, it all can be summarized in this sentence. The distinction between commensals and pathogens lies in their impact on host tissues, which I said earlier. Well, the next thing about innate immunity is that is the first line of defense against those pathogens. And our immune system, innate immune system, have different strategies of reaction to those pathogens. Our immune system can use avoidance, and this all can be, well, sorry for that. This all works because of existence of anatomic barriers like skin and mucosa, and also behavior modifications. These behavior modifications include some simple hygienic rules, for example, washing our hands and everything like that. Next strategy, which are quite important for innate immunity, it is resistance to those pathogens. Not always our immune cells are reacting to these bacteria and viruses as passionately as we sometimes can think about it. Of course, resistance, including just this cold reducing or eliminating of those antigens while they are in our bodies. The great role in this process had effector mechanisms had a great role in this resistance process, and those effector mechanisms, we can even say that they are suited to resist different categories of pathogens. Well, and the next type of strategy that our innate immunity can, well, trigger is a tolerance. This tolerance is not the same as the immune tolerance that can happen while our bodies are like trigger all these autoimmunity processes. This tolerance is slightly different. And this difference is that this process is just simply a response which can enhance tissue's capacity to resist damage that can be caused by those pathogens. So, to summarize the first reaction of our innate immunity, it's all started with those barriers. Then, well, pathogen are in our body. It needs all these complement and antimicrobial proteins. This is resistance. And then innate immune cells also here and present, and they are working against the pathogens and antigens, and they're after their work, the adaptive immunity can start. But this is the next topic. So we are stopping just at this innate immunity mechanisms. And all in all, first line of defense, innate immunity include anatomic barriers like skin and mucosal surfaces. They form an avoidance strategy and reinforced by additional resistance mechanisms. But well, nothing can be as good as it sounds. So if breached, those innate immune system, including complement proteins, responds immediately, contributing to both innate and adaptive immunity. And so pathogens and antigens are here. Well, and so our immune system don't need just to see them to react. Our immune system need to do something. And first of all, it's the triggering of inflammatory inducers. Inflammatory inducers are such a very specific type of molecules that can trigger that innate immune system reactions. Some of them are bacterial lipopolysaccharides, ATP, and urate crystals. After this inflammatory inducers, well, these chemical structures are, sorry, I'm really nervous. Sorry for this pauses. After that, sensory cells, they search for these inflammatory inducers in our bodies, and when they see them, they need to respond to them. And their response include the mediator synthesis. The mediators are specific types of immune system molecules, especially cytokines and chemokines. They are these molecules can trigger cytotoxicity and the elimination of these pathogens and chemicals inducers. After that, mediators, when released, they are flowing to the target tissues in which these inducers were spotted, and they start their work. For example, they're stimulating production of antimicrobial proteins or induction of intracellular antiviral proteins, or even the killing of some infected cells of the human body. The conclusion of that part is that the immune system can be activated by inflammatory signals, specific molecules and receptors, and they are indicating the presence of pathogens or tissue damage in the host's body. But innate immunity, as it was studied before, is not just only about molecules and mediators, it's also about cells. But those cells are not our usual T and B cells. This spectrum is much more interesting in terms of variety. innate immunity cells lineage started with common myeloid progenitor, or CMP, stem cells. This progenitor are the precursor of innate immunity cells like macrophages, granulocytes like neutrophils and eosinophils and basophils. Of course, there's also mast cells, which is full of granules with specific chemicals, and also there are dendritic cells that play a major role in responding to the antigen. All the cells are the part of innate immune system cell lineage, and they are very important in that part. So when we start to talk more about the cells, we can start with macrophages because they are, well, quite famous for their phagocytosis activity. Well, macrophages can be found in different tissues of our organism. They have a phagocytic role, so they're just like catching those bacterial or chemical elements and digest them to some extent. They also orchestrate immune responses. They can induce inflammation by phagocyting those pathogens, and they act as a scavenger, if putting it simply. Macrophages can also trigger phagocytosis, as was said before by me, and they can activate some bactericidal mechanisms. They also can play some role in antigen presentation and cytokine production. They have a different spectrum. All different tissue macrophages have different spectrum of slightly different spectrum of cytokines that they can produce to signal other cells that there is inflammation in this part of the body. Next type of innate immune system cells is granulocytes. There is three main types of them: neutrophils, eosinophils, and basophils. They name like all the granulocytes named like that because they include into them a lot of cytoplasmic granules that contain a lot of different molecules in them that play a major role in bactericidal mechanisms. Also, those cells, neutrophils, eosinophils, and basophils, have slightly different field of work. For example, neutrophils can induce mitosis, a special process of cell death that include those type of nets and molecule structures that can just coat those pathogens in them, and while that they can prevent the inflammation from going to some neighboring tissues. After that, neutrophils, they can die, of course. Eosinophils are extremely important in antiparasitical immunity because they granules contain a lot of specific enzymes that can cause well, the destruction of a parasite coat. And basophils, they produce a lot of inflammatory, they can trigger some allergic responses, and they can also work as an antiparasitic cells. And this is the main work for this type of the cell. And mast cells. This type of cell is quite unique because they are not that oftenly studied in like I'm just studied something different. So mast cells. These cells are extremely important in allergy mechanisms because they contain a lot of specific cellular granules, which contain mediators that can be released, for example, during allergic reaction. Also, mast cell can communicate with other immune system cells, and after that, they can trigger also some other immune system reactions. They are they maturing in tissues, after leaving the bone marrow. And also they can serve as an early infection sensors. They can contribute to the inflammation in skin, intestines, and our airways. Well, and another type of innate immune system cells is dendritic cells. They were discovered not so long time ago in 1970s, and they were derived from phagocytic cell class. Dendritic cells are the third class of phagocytic cell type, and they include some related to them lineages of cells, and their functions of those types of dendritic cells are still yet to be discovered fully. But anyway, we know them, of course, we knew the main function of dendritic cells. They can, of course, work as an antigen-presenting cells in our body. Also, dendritic cells, they can act as sensor cells. They can produce mediators, which can activate other immune cells. Initially, dendritic cells were recognized for activating T lymphocytes in adaptive immunity, but they also play a role in controlling responses of innate immune system cells. Well, but how all those mechanisms of recognition work? The work of those mechanisms is can happen because of pattern recognition receptors, and they can help with the main role of the immune system, distinguishing between self and non-self in the host body. And early observations showed us that purified antigens often didn't elicit immune responses without additional microbial components, and those components can be known as adjuvants. Adjuvants can help activate innate receptors on sensor cells, and those type of cells include macrophages, neutrophils, and dendritic cells. These cells can detect infections and innate immune responses by producing inflammatory mediators. There's a lot of different type of pattern recognition receptors, and the most famous of them is probably toll-like receptors. But it's not the only one. There is not only TLR, there is also ALR and MLR and some different. Those receptors recognize simple molecules and regular patterns of molecular structures of pathogens. Those structures are also known as pathogen-associated molecular patterns, and they are parts of many molecular microbial organisms. They can include mannose-rich oligosaccharides, lipopolysaccharides of bacterial cells, which is not present in human bodies. That's why, and they also have a highly conservative sequence of monomers in them. That's why our receptors, our pattern recognition receptors, can detect them quite well. Another part of innate immune system includes those molecules that can be produced by cells. These molecules, also known as cytokines and chemokines, of course, they're not specific only to the innate immune system cells, but they play a crucial role in their work. These are small proteins can be secreted by numerous cell types and, of course, innate immune system cells. Their main function is acting on the cells that secrete them, autocrine or nearby cells. These functions cause also autocrine and paracrine functions. They also can work in their local microenvironment, and they're specific because they are more local than systemic. Those cytokines can bind to specific cell surface receptors, and after that, they can activate a lot of intracellular signaling cascade, which can also trigger later reactions like phagocytosis, antigen presentation, etc. They can also play a role in opsonizations as well, which is and opsonization is a process of making some pathogens more visible and interesting to different type of to phagocytic cells.
Time. Don't forget about time. Sorry.
Yeah, I'm already finishing. Sorry. And chemokines are special types of cytokines that play a major role in inducing inflammation, infection, immune response. They also play a role in some more sinister processes like cancer, and they're especially important in innate immunity system responses. And other two types of cells are innate lymphoid cells and natural killer cells. They also play a major role in work of innate immune system. Innate lymphoid cells were discovered relatively not so long time ago, and their main function are still studied by a lot of scientists, but we already know they are playing a role in immunity to helminths and other parasites, homeostasis of epithelia, and development of immune system tissues. They also play a role in wound healing and maybe triggering of inflammation. Another studies were about connecting innate lymphoid cells with nervous system and the work of immune system in our brains and the role of innate lymphoid cells in brain-blood barrier, which is also extremely important part since there is a lot of autoimmune diseases that connected to the nervous system. And T cells or natural killer cells are part of innate lymphoid cells family, and they can also play a role as a like their role is slightly similar to the CD8 cytotoxic cells, but the thing is they do not have that much specific receptors on their surface. The receptors are more general and they can eliminate only certain types of pathogens, but nonetheless, if not them, our lives would be much worse than they now. And so, finishing my part of this meeting, during preparation of my, well, text, I was thinking about the possibilities in targeting innate immune system in therapy of different diseases. I met quite enough papers about cancer therapy that includes targeting some types of specific receptors on innate immune system cells. But there are also some studies about nanotechnologies and nanoparticles that can block those receptors, innate receptors. And I was thinking, but is that all? Is this therapy that efficient, that effective, or we already learn all about that we can now? Maybe some of you have interesting information of this specific type of immunotherapy. And with that, I want to finish my presentation and thank you all for listening.
Thank you very much. So yeah, if anyone wants to answer the question, feel free. And in papers, what information did you find? Like what cells, what type of cells in innate immunity did they target with nanoparticles? Like what did they affect?
Well, I found around 4 or 5 papers which were studying the nanoparticles with specific molecules that can target macrophages and phagocytose this process. And those papers were all made by Chinese authors. So I was interested in was these nanoparticle things more famous in other countries and in other like universities. Sadly, I didn't find that much information about this nanotechnology branch of innate immune system target therapy. And I also remember that some of those nanoparticles were like they having those receptor blocks in them, and the initial goal of this experiment was about delivering those particles to the cells and block the receptors. Genuinely, I was like thinking, but this is just like anti-something therapy, just a simple block. What's the toll with nanotherapy, nanoparticles? But eventually, I understood that this nanotechnology was not just something fancy, but this was all because they didn't want their nanoparticles to be like, how to say that, to be glued to some wrong type of cells or receptors. And this all was just about making this target therapy even more targeted. So that was the main thing that I found about that type of therapy.
Yeah, because it's interesting for innate immunity, because normally every therapy wants to target the adaptive immunity, like T cells. They want to activate either cytotoxic T cells or T regulatory T cells in cases of autoimmune diseases. So yeah, that's interesting. Thank you. Any questions, guys, to Ekaterina?
Well, I've read in chat one question that Elizaveta heard about dendritic cell-based vaccines against cancer. And I've also read about this type of vaccines, and I can say that one of the branch of innate immune system target therapy also include creation of vaccines of different types, but dendritics are most popular and the most well-known. And of course, the thing is that a lot of studies, a lot of from which I found was about cancer therapy. And the interesting part was about, as you said, Tatiana, that most of our current immunotherapy is about T or B cells, because they are not only the most popular, but they also play a major role in our immune system responses. But it also can be very useful for us to know better how cancer and innate immune system coordinate between each other, and how we can prevent some mechanism that can guard this cancer cells from the immune system, lessen their guarding ability. And I think there's a lot to think about and a lot to invent for our dear medical scientists. So we just need to wait and see.
Yes, absolutely. But not wait, maybe participate.
Or maybe work right now, which is much better.
Exactly. Well, any questions?
Good afternoon. Can you hear me?
Yes.
Yes, it's nice to meet you. I'm Alexei. I'm pediatrics resident from Moscow. Ekaterina, thank you for your brilliant message. It's really interesting. My question is not really serious, but according to the abundance of immune mechanisms and sophisticated system, sophisticated immune system, what's your favorite mechanism in the immune system, and perhaps what will be your interest in your future scientific career?
Well, if talking about immune system mechanism, I really favor the autoimmunity. I feel that I also can call myself like an amateur specialist in nervous system autoimmune diseases, how this immune system, for example, CD4 plus T cells, can breach through the blood-brain barrier and trigger some, for example, demyelinization processes. And the part that interests me the most is the process of, for example, a remyelinization and process of fixing everything that immune system broke during this process. I've also read some I've already read some papers and even experiments and preference about those autoimmunity therapy, and not just therapy, but rather mechanisms. How can our future therapeutic things, materials, not just drugs, work in the terms of preventing those autoimmune processes? The immune tolerance perhaps is the most interesting.
Yeah, Ekaterina, you can continue your answer on question. Sorry about that.
Oh, I was I used a lot of words, but in the end, I can always summarize all of my description in only one sentence, that I'm interested in autoimmune diseases and how our autoimmunity mechanism can affect our nervous system. And also another process that is very dear to me is also connects slightly to the innate immune system, about which I said earlier. This is the process of inflammation. So simply the aging of immune system, or another part, how aging of immune system affects the aging of the whole body. How all of those senescent cells can affect the immune system cells and vice versa. And I also say that this process of inflammation is part of my main interests of biology of aging. And I think it's not that famous, but quite popular in some circles. And I really want to explore it further as well. So thank you for your questions.
Okay, thank you. I hope you will achieve something reasonable in the future. Good luck with it.
Yeah, great. So now I think, guys, it's time to have another report. So next participant, please.
Hello, everybody. My name is Anna, and I'm an undergraduate pharmacy student in Sechenov University. So my aim for today's speech is to remind you basic principles of adaptive immunity. I'm sure that most of you is already pretty familiar with those concepts, so let's start. I'll begin with stating key points as a quick overview. Firstly, adaptive immunity is providing a protection from a vast array of antigens, whereas innate immunity only dealt with conservative molecular patterns. That's why each lymphocyte is ought to bear a unique receptor on its surface. Last but not the least is that cells of adaptive immunity are capable of immunological memory, which basically means that a subsequent response to the same antigen will be more effective. It is worth mentioning that colleagues on the Russian section a week ago touched upon research concerning innate immune memory, but this is more a question for a paper discussion rather than a classical point of view yet, but I thought that I would mention this. So let's move on. Basically, there are two major types of lymphocytes in vertebrate immune system, the B lymphocytes and T lymphocytes, B cells and T cells for short. They express distinct types of antigen receptors and have quite different roles, but most of them could be characterized as small cells with a few cytoplasmic organelles and a condensed inactive appearing nuclear chromatin, as you can see on the smear pictures below. As for basic terms, lymphocytes manifest little functional activity unless they're activated by the antigens, and they're called naive lymphocytes. After they have met their antigen and become activated and become fully functional, they are called effector lymphocytes. These two types of cells could be easily differentiated by receptors on their surface. B cell receptor resembles a basic antibody structure and often referred as MIG or SAG, standing for membrane immunoglobulin or surface immunoglobulin. So this antibody molecule is composed of two identical heavy chains and two identical light chain polypeptides. Heavy and light chain have variable and constant regions. Variable here are marked as orange, and constant region is blue. So the variable regions of a heavy chain and a light chain combine an antigen binding site, and here there are two of them. After antigen binds to a B cell receptor, the B cell will proliferate further and differentiate into so-called plasma cell. These are the effector form of B lymphocytes, and they will secrete antibodies. As for TCR, a situation is quite different. Thus, it also has a variable region, also marked orange, and a constant one, also marked as blue. It is composed of two chains, the TCR alpha and beta chains. They are pretty equal in size. The combination of these chains form a variable region, but here is only a single site of this binding antigen region. When a T cell meets an antigen and binds to its receptor, it proliferates into a broader variety of classes. It could be cytotoxic T cells, which are able to kill other cells, which are infected helper T cells, which provide signals or secrete cytokines and other effector molecules, and also regulatory T cells, which suppress the activity of other lymphocytes and limit the possible damage of immune response. So here you can see a full comparison table of both types of receptors. You can take a closer look maybe later. So moving to its functions, BCR and TCR ways of binding with antigen rather varies. Starting with the BCR, an individual antibody recognizes just a small portion of an antigen molecular structure. And this part is called an epitope. So typically, proteins have loads of different epitopes and could be recognized by either of them. Antibodies and B cell receptors directly recognize the epitopes of native antigen. But T cell receptors bind to a complex of an antigen, fragment and a self molecule. Here you can see a scheme. So T cell receptor can recognize epitope that are buried within antigens, not on their surface. Here it is marked red on the first panel. Then these antigens must be first degraded by proteases within the cell. It is shown on the second panel, and the epitope and peptide delivered to a self molecule and form a special complex, which is with MHC molecule, and a complex of peptide and MHC molecule can be recognized by T cell receptors. The right question to ask at this point is how this even possible to create such huge amount of receptors to each antigen that possibly an organism might face during lifetime. Not going into much details here, but there is a special mechanism which frankly amazes me every time I come across it. It is called VDJ recombination. So basically, it's a process by which T cells and B cells randomly assemble different gene segments together in order to create unique receptors. During this recombination process, the random addition or subtraction of nucleotides at the junctions of the gene segments create additional diversity, known as junctional diversity. So, of course, this great diversity of receptors has its own drawbacks and leads to certain hazards that must be avoided at any cost. That's why clones should be precisely selected afterwards as a safety insurance. So there is such a theory called clonal expansion, and these postulates are shown on the slides, but majorly cells should be checked that each of them has only one receptor specificity, and gene rearrangement process irreversibly changes the lymphocyte's DNA, and all its progeny inherit the same receptor specificity. That's why the proliferation of individual lymphocyte forms a clone of cells with identical antigen receptors. These processes mainly happen in the lymphatic system. The distribution of lymphoid tissues in the body is shown on the slide. Lymphocytes arise from stem cells in bone marrow and differentiate in the central lymphoid organs, which are shown in yellow here. B cells come from bone marrow, differentiating bone marrow, and T cells differentiate in thymus. They migrate from these tissues and are carried in the bloodstream to the peripheral lymphoid organs, which are also listed here and shown in blue here. They include lymph nodes, liver, spleen, lymphoid tissues, and let's take a closer look to the functions. The peripheral lymphoid organs are the site of lymphocyte activation by antigen, and lymphocytes recirculate between the blood and these organs until they meet their specific antigen. Lymphatics drain extracellular fluid from the peripheral tissues through the lymph nodes and into the thoracic duct. This fluid is known as lymph. It carries antigen taken by dendritic cells and macrophages to the lymph nodes directly. Here you can see an organization of a lymph node. It consists of outermost cortex and inner medulla, which is shown in dark pink. The cortex is composed of an outer cortex of B cells, and they are organized into follicles and paracortical areas made up mainly of T cells and dendritic cells. The medulla itself consists of strings of macrophages and antibody-secreting plasma cells, known as medullary cords. Naive lymphocytes enter the node from the bloodstream through specialized venules and leave with the lymph Another important organ is a spleen. It has no direct connection with the lymphatic system. Instead, it collects antigen from the blood and is involved in immune responses to blood-borne pathogens. Lymphocytes enter and leave the spleen via blood vessels. The spleen also collects and disposes red blood cells. Its organization is shown on the slide. Here you also can see different areas called white pulp and red pulp. The bulk of the spleen is composed of red pulp, which is the site of red blood cell disposal. As I mentioned earlier, the lymphocytes surround the arteriole are forming a white pulp, and the layer of lymphocytes around the arteriole contains mainly T cells. And also, we shouldn't forget about remaining parts of peripheral lymphoid system. So mucosal immune system or mucosa-associated lymphoid tissue, MALT for short, the gut-associated lymphoid tissue, and also nasal-associated lymphoid tissue and bronchus-associated lymphoid tissue belt. So to end my presentation, I want to say a few words about immunological memory itself, and it could be broken into simple statements which basically shows the sense of this mechanism. So lymphocytes encounters corresponding antigen in peripheral lymphoid organs. Then its clonal progeny differentiate into effector T or B lymphocytes, then can directly eliminate the infectious agent. and a subset of these proliferating lymphocytes differentiate into memory cells, ready to respond rapidly to the same pathogen if it's encountered again. So that's it for my presentation. After Ekaterina's presentation, I thought that it would be nice to discuss some of the trends in therapy targeting the adaptive immune system, because there is much more of this, and the spectrum is much wider, and the therapeutic agents have already been using for ages. So if you have something to share on this theme, it would be nice.
Thank you very much, Anna, for a presentation. And anyone questions or answering the question? So I can add something regarding the adaptive immune system, how to use adaptive immunity in order to generate some therapy. So basically, there are two strategies. Sorry, it's either you activate T cells, CD8 positive T cells, in order to kill cancer cells, or you activate T regulatory T cells in order to decrease the immunity, the strength of immunity, and to combat the autoimmune diseases. For example, system lupus erythematosus.
Maybe someone have worked with these therapies and can tell us more about it.
Well, so I can also add just a little bit about that. After listening this amazing presentation, I want to thank Anna for this presentation, and I also want to say that adaptive immune system therapy is, well, more famous than innate immune system therapy. But I was wondering about how CAR-T, or chimeric antigen receptor T-cell therapy, can it be implemented not only into oncology therapy like cancer therapy, but also into autoimmune diseases therapy? Because I saw several sources which said that CAR-T cells can be used while curing some neurological autoimmune diseases as well. Maybe it can also be used in other immune system diseases as well. So this is more of a question, yes.
So yeah, normally in CAR-T cells, you generate a synthetic receptor that targets cancer. So it basically upregulates the effector T cells, right? And I think the same way you can generate something that can upregulate T regulatory T cells. So you can target them.
As far as I know, I heard a lecturer a while ago. He told about the process of creating CAR-T. He told that it's really easy nowadays, and it's really it's very fast and very effective, and anyone can do it if he has right resources. But major problems are logistics to the patient, and if this problem will be solved, the production and targets have a really broad range of possibilities. That's why I suppose that CAR-T against the fight with autoimmune diseases is rather possible opportunity.
I've been working in a lab that was working on Tregs. I was working on the low dose interleukin-2 to activate Tregs as my master 2 thesis. And another team in my lab, they were working on generating CAR Tregs, basically. So they modified, as far as I remember, the receptor in order to target something, I don't know, the tissue or the organ, using this Tregs. So they basically reprogrammed this Tregs in the lab, and then they that allowed them to target the specific tissue, with autoimmune disease. But yeah, it's very interesting, the CAR Tregs. So maybe you can search something regarding CAR Tregs.
Great. So my name is Anna. I'm an undergraduate student. I'm pursuing my third year, and I mainly major in molecular biology, and I did a bit of immunology. So it's quite an interesting topic for me. Can you see my screen?
Yes.
Yes.
Wonderful. So having heard and talked about the innate immunity and the adaptive immunity and all those mechanisms, we would like to piece everything up together. And this is where the effector modules in our immunity come into hand. And we know that as we talked about the different pathogens, especially Ekaterina mentioned this in the beginning, that we have indeed different lifestyles. And for those different lifestyles, we indeed have different effector modules for our immunity to target them and tailor the immune response to them. And we can talk about the intracellular viruses or bacteria and protozoa that mainly live and reproduce within the cell. And therefore we would like to target and activate the cells that would be those infected cells, or that would neutralize the intracellular pathogens. On the other hand, we do have the extracellular pathogens, the bacteria, helminths, other protozoa. Here on the picture, for example, I gave an asteroid helminth. And for those purposes, we would have absolutely different effector module that would probably target the entire pathogen, that would be cytotoxic to the pathogen, not the cell, or that would engulf the pathogen solely by, let's say, macrophages or mast cells. So to wrap it up, the effector module that I'm going to be talking about is basically a collection of the cell-mediated immunes that interacts with the cell and recruits different cells, and the humoral mechanisms, mainly the antibody mechanism, that involves the components of both systems, the innate immunity and the adaptive immunity. And we're going to be seeing them interacting with each other, piecing up together. And these mechanisms, they act together to achieve the elimination of the particular category of the pathogen. And in our case, we have to be thinking about these categories of pathogens that could be intracellular, such as viruses, or bacterial pathogens, and that could be extracellular, such as helminths and other bacteria. And therefore, we can arrange the effector modules into 4 different subsets. And depending on the nature of the pathogen, different adaptive immunity cells and innate immunity cells are going to be activated. We mainly focus on those effector modules, talking about the activated T cells. Whenever it comes to the cytotoxicity response, we tend to have the cytotoxic T cells activated. Those are the T cells that express CD8 receptors. We're going to talk about this later, or natural killer cells that are also stemming from the lymphoid progenitors. And they act on elimination of virally infected or metabolically stressed cells. And also, we mentioned them before in terms of the targeting the cancer cells. And later, we can tear apart some of the effector modules into 3 types. The first type involves the activation of the T helper 1 cells. And also, as Ekaterina mentioned, there are innate lymphoid cells, and they also have classes. And in this case, for the type 1 immunity, we activate the ILC1. And they act upon elimination of intracellular pathogens such as viruses, and they do activate macrophages. And mainly, their main profile is enormous secretion of interferon gamma. The next types that we're going to see in this presentation is the mucosal and barrier immunity called type 2. It recruits the T helper 2 cells, and they mainly target the elimination of parasites, and also they recruit the cells of innate immunity, the polymorphonuclear granulocytes, such as eosinophils, basophils, mast cells. And another type of the extracellular immunity is type 3. It also has its own profile, the cytokine profile, and the profile in T cells, and it mainly recruits T helper 17 cells and work on elimination of bacteria and fungi that do replicate on their own within the tissues. So talking about all those effector modules, we do have to understand that everything works interchangeably. And upon arrival of a pathogen, we do have the antigen presenting cells being activated that then activate the T cells. And here mainly the T cells overall orchestrate the cell-mediated immunity. We've already mentioned that the T cells could be divided into 2 different types, which is the ones that express CD8 receptors and the ones that express CD4 receptors. And they are mutually exclusive. There are no cells that can express both if it's not a defect. These receptors recognize the different classes of the major histocompatibility complex that is expressed on each and every cell in our body. We got 2 classes of MHC receptors, the first class and the second class, and they are expressed in the different cells. And CD8 T cell recognizes the MHC1 class protein, while the CD4 T cell recognizes MHC2. We're going to talk about this a bit in the detail. So I said that MHC receptors are expressed differently on different cells. The MHC class 1 receptor is expressed on the surface of each and every cell in the body, and it is, in our case, important for targeting viral infections. Whenever the virus infects the host, whenever the virus infects the cell, it is either broken down or mainly it is replicated in the cytosol. The replicating fragments, they start to form in the grooves of the MHC receptor, and later they are exhibited in the surface. So they're expressed and then exhibited on the surface of the cell. This peptide, mainly the viral peptides become trapped in the groove. And it is later targeted to the T cells that would later recognize the MHC receptor, and this interaction of MHC receptor and the T cell receptor would activate the either cytotoxic response. In our case, as we said, in a second. As we said, the MHC class 1 receptor is recognized by CD8 T cells. So whenever the CD8 T cell is activated by the MHC class receptor and also other 4 receptors and have all the other cytokine profiles around, it exhibits the cytotoxic activity. In another case, as we can see here, whenever it recognizes, it kills the cell. So the T cells can control the infection by directly killing the infected cells before the viral replication, or just after the viruses are released. Yes, and as I said, the MHC class 1 molecules are mainly expressed on most of the cells of the body, and therefore they serve as an important mechanism for antiviral defense, as any cell could be recognized, any cell being infected could be recognized by the cytotoxic T cells. Another mechanism that is exhibited by the T cells is the T helper response. And it is another effector module. So the CD4 T cells recognize another class of the MHC receptors. It is the MHC second class. And it is only expressed by the antigen-presenting cells, such as the dendritic cells, macrophages, and also B cells. So the antigen-presenting cells tend to sample the tissues, sample the fluids around them, and break down any pathogens that they encounter to later later create the pathogen and bind the peptides of those pathogens into the MHC second class receptor to then exhibit those MHC class receptors at the outwards. And the CD4 T cells can recognize those peptides on the surface of the antigen cell are activated. They could develop into a variety of different effector subsets. Those are mainly the T helpers first class, the T helper second class, and the T helper 17. In the lymphoid tissues, a subset of CD4 T cells, also those that reside in the lymphoid tissue and not dispersed around all over the body in the fluids, is called the T follicular helper. It mainly interacts with the B cells and activates the B cells to regulate the antibody production during the immune response. So those effector modules be later split into the differentiation into T cells that circulate in the body fluids, and those T cells that are residing in the lymphoid tissue and the germinal centers. And also, apart from the activation, the direct activation of the cells and the cytotoxicity, the T helpers can secrete the essential cytokines that are very important for the further immune response. Here we can talk about the first type of immunity, is the T helpers produce large amount of interferon gamma, and we tend to say that interferon gamma activates macrophages and increases the intracellular killing power, and it is mainly achieved by the fact that whenever the macrophage is infected, and here we're going to be talking with the example of the mycobacterial infections. Those are the intracellular parasites, and they do survive intracellularly because they prevent the fusion of the vesicles, and they prevent their degradation, the further degradation, whenever they are engulfed by the macrophages. So lysosomes are not fusing with the phagosomes, and therefore mycobacteria resist and are maintained there within the macrophage. The antigen-specific T helpers, whenever they recognize those infected macrophages, as you can see on the picture, they start secreting this interferon gamma, and the interferon gamma breaks through the work of diffusion of the lysosome, helping macrophage to disrupt the intracellular pathogen. Also, T helpers 2 and T helpers 17 also produce other cytokines that are specialized for promoting responses against extracellular parasites, fungi, and helminths. And this is just a very short shortcut, a very short short of how it all pieces up together. The next slide. Also, as I said, T helpers, our immunology tutor says our T helpers give a pat on the back to the B cells to activate them. And whenever the B cells are activated and are differentiated into the B plasma cells, they start secreting the antibodies. And the antibodies are the key players in the protection against especially extracellular pathogens. So they are mainly resembling the pathogen-associated patterns there. There are 5 main forms of the antibodies, and those forms are called isotypes. The first undifferentiated type, before the isotype switching happens, is the IgM. You can see here on the picture, it has the star-like form and shape. It is very good in terms of, it is very non-specific, but it's good in terms of agglutination and binding to pathogens. However, whenever we have some specific pathogen, the class switching happens and the antibodies become more tailored. They are expressed in different forms and shapes so that they are tailored upon the pathogen lifestyle. And each class carries out its particular function by engaging in a distinct set of effector mechanisms. So for different effector modules, we have different types of antibodies. We can talk about antibodies eliminating the infection in 3 different paths. The first one is just direct neutralization. Direct neutralization is whenever antibody meets the toxin, encounters the toxin in the blood, on all the body fluids, it just encompasses it all, it binds to the toxin and neutralizes it mainly, so that the toxin can no longer circulate in the blood flow. And later, this agglutination complex is ingested by the macrophage and all the toxins are neutralized. So the macrophages on their surface have the receptors that recognize the specific antibody. Wonderful. I'm probably going to continue. So we talked about agglutination and the direct neutralization. The next path with the next path for antibodies to interact with pathogens is opsonization. Opsonization is a term for the same kind of encompassing the pathogen, but in this case, it's not a toxin, but it's the entire cell sometimes, to make it more attractive for the macrophages. So basically, the antibodies bind to the surface of the pathogens and recruit by this binding the macrophages, mast cells, are the phagocytic cells to neutralize, or neutrophils to neutralize the pathogens. And also, antibodies against pathogen is the essential element for activation, the third type of the complement pathway. And complement pathway, once again, pieces up together the innate immunity and the adaptive immunity. So it's very multifacial, it's very multifunctional. So to wrap up everything, we've talked about the orchestration of the different events that come upon the arrival of a pathogen. First, we have antigen-presenting cells that then secrete some chemokines, secrete some interleukins. Those interleukins activate the T cells. T cells orchestrate the entire factor modules. They recruit the B cells, they recruit the macrophages, and everything happens very interchangeably. Everything is very fluid in terms of the response. However, we talk about this as a very precise system, but sometimes we can encounter inherited and acquired immunity defects that can lead to the lethal or just partial immunodeficiency. And we all know the perfect example of the acquired immunodeficiency, the very bad example, is the acquired immunodeficiency that stems from the HIV virus. HIV viruses, HIV targets mainly the CD4 T cells, which leads, and as we can see, the CD4 cells, the T helpers, they are the essential players in the effector modules. And whenever the neutralization and whenever the infection of the CD4 cells happens, the immunity can no longer orchestrate this so finely and so beautifully. And therefore, our immunity with the immunodeficiency syndrome happens to be very exposed towards the intracellular bacteria and other viruses. So many patients with acquired immunodeficiency syndrome, they die not due to HIV, but mainly the exacerbated effects of other diseases that cannot be targeted with CD4 T cells. And the best way to eradicate any type of the diseases by this time is the vaccination. And here we can talk about the effects of vaccination. On the example of the States, they claim to completely eradicate diphtheria, polio, and measles. And also together with measles, there's a brain disease called the SSPE. It stands for subacute sclerosis panencephalitis. And we can basically see the entire shutdown of the disease 20 years later after the vaccination was administered to the population. However, we also have to mention that even though some of the diseases could be eradicated in some countries, the immunization still have to be maintained worldwide, because especially nowadays, the epidemiological circumstances are very fluid within the different countries. And therefore, the vaccination must be administered in each and every country to actually prevent reappearance of eradicated diseases. Thank you so much for your, thank you so much for listening. I didn't add a slide that would promote thinking, but I'd like to actually talk about, so we mentioned those targeted therapies. And in my case, that would be very interesting to talk about actually the evolution of the adaptive immunity and how come all those cells just came together to play such a vast role in the effector, in the effector modules. Ekaterina in her presentation mentioned the innate lymphoid cells. They stem from the lymphoid progenitors, even though they exhibit the innate functions. So there were loads of speculation after the discovery of the innate lymphoid cells that those are the intermediate state for evolution of our current adaptive immunity cells, for our T lymphocytes and the B lymphocytes. And some of the innate lymphoid cells carry genes to express the antigens. However, also natural killers, they have the genes to express the antigen receptors. However, they are not expressed and they're just inhibited by some of the transcription factors. So it could give us a hint into the evolutionary development of the adaptive immune system and how it all then just play together with the innate immunity to neutralize the pathogens. If you have any ideas on that, that would be very interesting to discuss it. And anyways, I'm just open to questions on the presentation.
Thank you very much for a wonderful talk. And anyone who wants to take part in the discussion or to answer some questions, feel free.
Well, I want to thank Anna for her amazing presentation. And I don't really know the exact answer to the question about evolution of adaptive immunity and effector systems, but to continue the topic of innate lymphoid cells, I've read several points about them and their interaction with nervous system, in which there was stated that different types of innate lymphoid cells, ILC3 and 2, if I'm not mistaken, they were affecting the processes in nervous system differently. For example, some of them were pro-inflammatory and some of them were anti-inflammatory cells, well, acting like that. And I was just thinking, like, how can it be that almost the same population of cells can develop like these two distinct branches of cells that can, well, trigger the different reactions in the same place, because I was used to thinking that there is different types of cells for different mechanisms, or there's only one type of cells. They can manage to act as pro and anti-inflammatory cells. And this is just like two types in one family that with coexisting, they actually antagonistic, and they also, all of them existing in the nervous system. Sadly, I didn't manage to find any exact information to this topic and how it happened, how it was like that. But it would be interesting to maybe learn more or discuss this thing as well, or not just discuss, but think about this and all those effects of innate lymphoid cells on immune system, for example.
So ILCs, they are indeed very fascinating because they not only branch into two different subsets, there are indeed anti-inflammatory and pro-inflammatory profiles that they secrete, but there are actually three types of the ILCs, and they in some way resemble some of the T helper cells and their cytokine profiles. So I guess their inflammatory function all depends on the chemicals they secrete mainly. But also, yes, they are very multifunctional and multifacial, which is very fascinating.
So for the evolutionary mechanisms of adaptive immunity, that's really interesting. Have you found some information? If you began the talk, maybe you are aware of some evolutionary mechanisms of adaptive immunity so far, so you can tell us more what you have found already, or
yeah, the only speculation that was given, and probably I might find a bit more from different recent papers, but there was the gene duplication in some of the clusters that happen in the lymphoid progenitor that firstly allowed the differentiation. So basically those genes that are acting upon expression, that the genes that code for antigen receptors, they used to be the other genes that were expressed in the innate lymphoid cells, and after the gene duplication, they came to serve the other functions, and they gave rise to the other population and other lineage of lymphoid cells. Whereas in the innate lymphoid cell, the gene just retracted the function, and basically the function was deleted for the antigen presentation, for the antigen receptors. But it's also, everything is very intertwined. It is quite hard to wrap my mind around it.
Yeah, but it's a good idea to suggest a paper for a meeting. So yeah, that would be
that would be wonderful to read something.
Yes. Okay. Any questions to Anna? Otherwise, we are well in time, so we can finish now. So Yes, it's your last chance to have a question.